Oh, man, I've had enough of doing year 10 science. I think I want to do some year 11 biology work. But what should I do? Hmm. What's that? Yeah, that in the microscope. Sounds good. Okay, the first thing we're going to look at is the root. Now, this is a root tip that's undergoing mitosis as well, which is something that's going to be really interesting for us going forward, and something that's quite pertinent to looking at roots and looking at this idea that we're starting to get some differentiation, cell specialization in this area of, let's call it, lots and lots of activity for a plant. Now, if we look at the top here, so like we did there in the intro, this is investigation 5.1 and it's on page 137 and 138 of your book. Now, what we're going to start to see is that here at the top of the root, we'll call it, closest to the stem, we're starting to see this area of maturation. As in, there's lots of mature, fully differentiated and fully specialized cells in this region. And as I move down it, we're starting to see that it's starting to get a bit more densely packed together. And this is the area of elongation. The area of elongation, right? Where we're starting to get some more differentiated cells, more or less of this apical meristem, which we see here in the tip at the bottom. Lots, really, really densely packed, and especially down here, no differentiation. And we're going to zoom in now and actually see that difference. Okay, so here at 400 times magnification, I'm right at the tip of the root now. You're seeing lots of undifferentiated cells. They're not really very different from their neighbors. They're not doing anything different then if we're thinking about this idea of structure and function. And as I move along it, you can actually see, which is really quite amazing, especially this cell here, this cell here, this cell here, that one there, these couple here, all of the black sort of dots and stuff that you're seeing, and these like little looks like a bunch of like kind of fingers or some grass or something growing out there. That's actually some chromosomes, some condensed DNA that's actually undergoing mitosis. So each of these cells is actually undergoing mitosis, or is at one of the various stages of mitosis. It's not something you need to know just yet, but it's something that's very important for you to understand, that if we have lots of cell replication going on and nuclear divisions like mitosis going on, it means that these cells are just rapidly forming, replacing themselves. And it makes sense, you know, if a root is growing down into the ground, it's spreading out, it's increasing its surface area physically in the ground, and it needs to replace all of this tissues that's working through the ground, being pulled apart, being eaten by bugs, all those things. But these cells don't actually have a particular function other than growth, which is really what mitosis is doing, which is actually really quite cool. And then if I keep moving along, this might be very hard for me to drive here, but I'm going to try. And you can see as I move along, less and less mitosis is occurring and more differentiated cells. These cells are becoming elongated. We're in this area of elongation. I'm going to try and see if I can get back. Yep, there we go. This area of elongation. And as we move further up, now you're starting to see these cells are really well defined, less mitosis going on, much bigger cells doing a job now. And their job now is becoming the vascular bundles, the xylem and phloem, that actually have this job of moving the products of photosynthesis and the reactants of photosynthesis, as in the water and nutrients required. And now let's have a go at looking at some stem cells. Now we're looking at the structure of some stem tissue. Actually quite a nice one. This is so your investigation 5.1 calls you to look at a young stem tissue. We're going to do that. This is a mature one, and I think it just leads well into some of the other stuff that we're going to be doing. So you can see clearly defined epidermal tissues. You can see this layer here forming like the outer layer, the waxy cuticle, I guess, so that we're not losing too much gases and water out to the environment. We've got our ground tissue, which is all of this stuff here, made up of parenchyma cells. When we're starting to look in here at the part of the vascular bundle, which is this section here, the xylem and the phloem. So it's nice that you get to see this actual a real change here from the roots, that there's actually some cells that are really doing some very specific jobs here. And you're starting to see that differentiation in structure, which means that we're getting a differentiation in function, which is quite nice. But if we're going to describe the difference here between those two, we're seeing these very defined cells. What I'm wanting you to do is to do some labeled diagrams for all of these things, but also, you know, in the discussion, thinking about how is the root actually maximizing its surface area. Why is this important? Surface area both in root hairs and in terms of spreading out into the ground. What's the function of different types of tissue in this transverse section of stem? And then relate the structures viewed in the root and stem to their functions in obtaining and transporting nutrients and in gaseous exchange. So quite a lot there, but actually just knowing the structure of things allows us to realize that there must be a different function associated. I'm going to flick over to the young root cell, keep getting the two mixed, the young stem cell. tissues, sorry. Man, all over the place. I need a script or something. So here is the young stem tissue, young stem tissue, still starting to see this epidermis layer. The ground tissue hasn't really fully differentiated yet, because there's still some stuff going on in each of the cells. Looking at growing still, remember it's a young bit of stem tissue. And here you can see one dedicated vascular bundle of phloem and xylem. So we haven't even got as many of these vascular bundles. As it gets bigger, as it gets larger, the requirement, more leaves, it's going to require more of movement of nutrients to start doing its job and to look after a bigger plant. So this idea that we're starting to see differentiation here, for certainly, but nowhere near as much as in the mature plant. So even that comparison between a mature plant versus a young plant, and we're starting to see some differences there, which is quite interesting and cool. So have a go at investigation 5.1. That's pages 137, 138 in your textbook, with the associated stuff on Schoolbox. Again, thanks for watching and good luck.